Thanks so much. It's a pleasure to be here. What I thought I'd do is give you 3 examples of things that we're doing, which we're doing here at the Koch Institute. And really, they're aimed at several things. They're aimed at solving, I think, big international scientific problems. And also, they are leading to companies as well, because that's helped us do that. Let me just start right away and say the first one. Actually, maybe I will steal the microphone so I can walk around.
You have a lapel on.
Okay, so does this work? Can people hear me all right? Okay, great. So the first one actually is something I've thought about for a while, and I've had the fortune of having a fantastic gastroenterologist in our lab, Gio Traverso. He actually is sort of a picture of Convergence. He got his PhD with Bert Vogelstein, who's an outstanding molecular biologist at Johns Hopkins, and he's a card-carrying gastroenterologist. And this whole project started about 4 years ago when Bill Gates came to see us and was talking to me about some of the big problems in the third world. And of course, some of the problems, as you'll see, they also affect our world too. But in particular, one of the terrible things in the third world is patient compliance. And one of the things that we wanted to do is create pills. Right now, whenever anybody here takes a pill, you take one a day, maybe one on the second day, and so forth. What we wanted to try to see if we could do is, could you ever take a pill once a week, once a month, maybe even once a year? And is that possible? Well, first, let me just go over some of the issues about people taking drugs, period. And this is from the World Health Organization. These are just some direct points they're making. First, poor adherence, they say, to treatment of chronic diseases is a worldwide problem of striking magnitude. Adherence to long-term therapy in developed countries like ours, that's 50%. Just as an example, Denny Ausiello, who's a friend of mine, he used to be chief of medicine at Mass General. He said, give you a quiz. He said, what do you think is the mean amount of time somebody takes a statin for? You know, you're supposed to take it for the rest of your life if you have heart disease. What does anybody think? 3 months. That's the answer. People are pretty pathetic, but that's what happens. Basically, in developing countries, it's even lower. The impact of poor adherence grows as the burden of chronic diseases grow worldwide. And they point out that increasing the effectiveness of adherence interventions, that may have a far greater impact on the health of the population than any improvement in terms of a specific medical treatment. The New England Journal of Medicine says similar things. Half of all patients, don't adhere faithfully to their prescription. The result's $100 billion. This is 2010, on avoidable hospitalizations. And it's estimated that better adherence to antihypertensive treatment alone could prevent 89,000 premature deaths in the US annually. Well, will the idea that I'm mentioning, taking pills longer, help? Well, here's a study from Japan where they actually show with certain drugs that if you look at persistence, if you do daily oral dosing, you see this kind of drop-off. But with the same drug, there's a drug that you can make very long-acting just because of its chemical nature, alduranates. But if you do weekly dosing, you do better. If you do monthly dosing, you do better still. Obviously, I'm sure you don't need me to show you that if you go from 4 times a day to twice a day to once a day, of course you'd see the same thing. But I think this is a little different. This hadn't been done till last year. So what's the challenge? How could we ever get drugs to last more than a day? Well, that's not so easy because you swallow a pill, it goes through in a day. People have tried a variety of ways. So first of all, what does a standard capsule do? I didn't make this, the students do, but that's pretty typical. There's been a lot of work over the last 40 years of doing what I'll call bioadhesion, making drugs adhere a little bit better, like mucoadhesion. And that kind of slows things down. But still, if you look at it, it doesn't go much longer than a day. Our thought was, could we ever swallow a pill that will actually, when it gets into the stomach, open up to some giant object, but very, very open, that will just stay there until we make it degrade. But while it's there, it could deliver the drug. So let me do this one. And so the idea is that if we could actually make this last different lengths of time. So anyhow, how could we do this? What are the challenges? Well, we thought there are 5 challenges. Can we control drug release rates? Can we deliver a drug for a long period of time? Can we control polymer degradation? So just to go over what the idea is. So I'm going to give you a capsule. But in that capsule, we're going to have some object that once that capsule dissolves, and we can make the capsule dissolve easily because all it has to do is get in the stomach, which has acid. But once that capsule dissolves, we've got this thing that we're going to put in, but it's a polymer, and it has a variety of properties. One of the properties is it's got something like shape memory or superelasticity, so that once the outer part dissolves, it just opens up. That's the idea of controlling polymer shape. We also want to have it be very, very open so food can pass. And we also don't want it to stay there forever, so we want to be able to do it degrading. And finally, of course, we want to be able to do it safely. So let me go over each of these challenges. Can we control drug release rates? That's something we did a long time ago. We started back in the early 70s, and that was sort of some of our initial contributions that led to the stuff on the wall. And we created little microspheres. Here's one cut in half. And the way we can control release rates is if you look at a section of these, what we can do is we can put drugs in these. This is a cross-section. But the drugs will slowly diffuse out through tortuous pores that we make in it. And this is an example of the last slide, but a year later. And so we can make pores, and by controlling the porosity and what I call the tortuosity, the windiness of paths in there, we can make these last almost any length of time. So how do we control release rates? Well, we can change the particle size of the drug we put in. In other words, if the particle sizes are big, the pores are big. And then your release is fast. If your particle sizes are small, pores are small, release is slower. This is just a simple example of that. Similarly, we could change the loading. That's the amount of drug. And you could imagine, again, one analogy I use for delivery is like the streets of Boston. If you have a lot of interconnections, you will get out faster. So if you have a high loading, meaning a lot more drug, you'll have a lot more interconnections, you'll get out faster. And this just shows that. Sometimes you can do things like coat them. That'll make it slower. So we have a whole bunch of ways of controlling it. Even the polymer molecular weight allows different release rates. Literally, by changing these parameters, loading, coating, particle size, molecular weight, you can get 10 to the 5th different release rates. I mean, really, five orders of magnitude. So you have enormous ability to control release rates. Can we deliver drug for a long time? Well, really, the way we deliver drug for a long time is we capitalize on what I just told you. We could pick small particle size, low loading, high coating, high molecular weight. And this is what's called against the square root of time, which means 140 hours is literally 140 times 140 hours. That means we've released this from a tiny little system for over 2 years, if you want. And we could go longer, but the students usually don't like doing experiments for that long because if it doesn't work out, they have to stay a long time. So we can do that. And all this has already led to systems today that are widely used by many millions of people, but all these are injected underneath the skin. And there's all kinds of systems, like ones that can deliver, that are, like, for example, this is a once a month, now once every 6 month injection for prostate cancer. This is a once a week injection for type 2 diabetes. These are more rapid ones for preventing bleeding. This is a once every 2 week system for preventing schizophrenia. This is a once a month one for preventing narcotic addiction. And so anyhow, all these are already on the market, widely used, but they're all injectable. So you could imagine that the compliance issue is not as good. We want to be able to do it orally. Also, I said, could we control polymer degradation? Well, here we developed a trick where what you can do is make what we call copolymers. And copolymers, unit A, unit B, and they're connected by bonds like anhydrides or esters that are degradable. And by varying the ratio of this to this, what we can do is make the polymers last anywhere from, say, 2 weeks. This will last for about 4 years. And we could make it even more. But the next one is, could we control polymer shape? Because remember, when we compress it into that system, when it comes out, we want it to assume a shape. So we've published several papers in Science and Nature where we showed that you can make polymers with what we call shape memory, and by simply changing the temperature or in other cases, shining a light. We have different chemistry we build in. We can actually make them change shape. Let me just show you an example of that in a second. So what we did, what I'm going to show you, and we originally thought about this for minimally invasive surgery. So I'm going to show you a string at room temperature air, and we're going to drop it into water, and it's going to change to a coil like a stent. At least I hope it does. So here it is, room temperature air, drop it into body temperature water, and it assumed that shape. You can even make knots tie themselves. Say you have a surgical suture. Say you have a wound inside your body. We could actually make a knot tie itself. For example, here's the loose knot at room temperature air, but when it goes into body temperature water, it just tightens. So we can do that as well. So finally, can we do all this safely, effectively, and relatively inexpensively? So what Gio and his team did is we've actually modeled different shapes that we want our capsule or the stuff inside the capsule to assume once the capsule dissolves in the stomach. One such shape is this. Another such shape is a star. And why we like these shapes are they're so big, they can't pass through that pylorus. That's the part between your stomach and your GI tract. They'll just sit there. But they're so open, food can pass. And like I say, we can make them degrade and release at any length of time. Here's some pictures of those. And there's all kinds of ways to engineer them. So let me just go over one of the things that we've done with the Gates Foundation. Ivermectin won the Nobel Prize last year in chemistry, and they feel that that could be a great way to stop malaria. And Phil Eckhoff, who works with us closely, and Hannah Slater, they've all basically shown that if you can keep the level of ivermectin in the blood for over 2 weeks, over 6 nanograms per mil, that should get rid of malaria. I guess it's theoretical. But that's the question. Could you ever come up with one pill that you could just swallow and do that for this whole course? So Gio and his team have done that. This is what you normally get with ivermectin. But here you have 3 pigs. This is with the star. And for 16 days, you're above that level with these pigs. They've done the same thing. This could be for TB. This is a once every 10-day pill. And of course, maybe another question to ask is it safe? So since they're gastroenterologists and clinicians, they've done over 200 pig experiments, no adverse effects, no change in appetite, no obstruction, no perforation, no change in weight or stool frequency. And then, they did endoscopic evaluation, and they didn't see any injury there. So this is one thing that we've been working on, and we formed a small company to actually develop this for the first world. But what's exciting is that we might really be able to wipe out diseases like malaria, possibly Zika virus, with these pills that people could take for long times. Second area is actually making a system smart. I had this idea a number of years ago, and here I've worked closely with Mike Sema, who's one of my colleagues in the building. Could we make a microchip that we could put different drugs in? And here we have these caps. They could be made of a metal like gold that we can make dissolve. And in each of these wells, we could put the same drug or a different drug. You could literally have a pharmacy on a chip if the drugs were potent enough. So we've actually made some. This is an early chip. It's basically, this is a top view, this is a bottom view. And this is a United States dime. By the way, this is a short, flat chip. They don't have to be like that. We can actually make cylindrical chips you can inject through like even a 20-gauge needle. Here's how they work. You have a well. It's covered with a metal like gold or a platinum alloy. It'll stay like this in the body for years. But if you apply 1 volt, like with something like this, off comes the cover, and the drug comes out. And this we published in Nature. And you see different amounts of drug coming out from different wells at different times. But if you wanted the pharmacy on a chip idea, you could do that too. Those are different amounts coming out. And then what we've actually done is take this into patients. It wasn't easy. Again, we started a small company, Microchips Biotech, and John Santini, our student, led it for a while, and now Cheryl Blanchard is. And basically, they went from small animals to large animals to humans. And a couple years ago, we published this, where we could put the chips in the human body. You can actually communicate with them over a special radio frequency called the Medical Implant Communication Service Band. It's approved by both the FCC and the FDA. And you can even, if you're worried about tampering or if the patient wants to change it, you can have a special computer code to administer or change the dose. And there's even a bidirectional communications link between the chip and the receiver. The receiver, by the way, could be something like this, or it could be an iPhone. Depends on what you're trying to do. And it tells you, did you take the drug? You know, again, as I get older, I keep thinking that's very important to know what I did, and battery life and so forth. The example that we did the first trial on was 8 patients. And I should say, usually when we do these things, people are very good, like the review study sections, are very good at telling you why your idea won't work. And the number one reason they told us is you get fibrous encapsulation. So therefore, a molecule wouldn't get out. So we said, let's try a really big molecule. If that gets out, smaller ones should get out. So we tried parathyroid hormone, which is 39 amino acids. We also chose it because we felt it could do some clinical good. Right now, when women, like say in their 70s, they have osteoporosis, which is a bad disease, and they're supposed to take an injection of it once a day. But 70% of the women drop out. They don't do it. I mean, nobody wants to inject yourself once a day. So what's done, this will just keep doing it for you with one dose. So what we did here is a small office procedure for the implant. And what we saw with the same pharmacokinetics, that's the release curves, I'll show you that in a second, with actually less variability than injections. And these are 3 measures of how well you're treating osteoporosis, and they're the same. Here's some of the human data. This is actually the release in the body at days 60, 68, 76, and 84. They're pretty much superimposed. And you do get a little encapsulation. This is the chip with actually, the apparatus actually is a chip. There's actually a computer program in there, microelectronics, a power source, and we even imprinted an antenna into it. That's how you communicate with us. It's not like just opening up a garage door almost. And basically you get, this is histology, so there's no inflammatory cells. So this is one project. Another thing that we're thinking about doing and is someday is making smart systems where you could actually put sensors on these chips so the chip could detect stuff and then deliver in response. And a whole other project that came, which is very interesting, as I mentioned, again, we do a lot with the Gates Foundation, and Bill and his colleagues came here and they said, well, you know, one of their big concerns is the third world. Right now, women, the birth control is very poor, and there's an enormous number of abortions, often causing death. So right now there is birth control available, but it's not very good. It never lasts for more than a few years. And secondly, if the woman changes her mind, there's no way to turn it on or off. They said, is there some way you could give a little system that might last for, say, at least 10 years, and then somebody could turn it on and off anytime they want? In other words, could you have a personal fertility control system? So they gave us this grant, $13 million so far, and this is actually right from their website. It's a personal fertility control system with emphasis for use by women living in developing world countries as a means to effectively plan their families. And a way to think about it is we have a tiny little chip. It's got 200 wells. Each well has enough drug for a month. And what you do is the program's set, and the first cap comes off, that gives you the first month. Second cap comes off, that gives you the second month. Let's say somebody changes their mind. They press a button like this, the third chip doesn't dissolve. Say a year later they change their mind again, well, they press the button and now it will, and then and on and on. So this is another area that we've been working on. And just to show you what it looks like, here is an example of a chip. And the very last example that I want to mention is, it was very interesting. It was also a very good example of convergence. I had a really good graduate student, Armin Shari, who was from Stanford, and he worked jointly with Klaus Jensen, who's an expert on microfluidics, and myself. And one of the things that we began looking at is, could we insert materials into cells? And right now, there are a variety of ways to do it. Electrical ways, cell-penetrating peptides that poke holes, nanoparticles, and so forth. But they all have problems. Like electroporation can cause damage or toxicity. Cell-penetrating peptides cause chemical modification. And the nanoparticles often get trapped in the endocytes and so forth. So he was playing around one day with ways of injecting things, but with, like I say, Klaus is an expert in microfluidics, so we actually looked at, could we pass a cell through this tiny little channel and squeeze the cell? And as I'll show you, the ability to do that, which we can do safely, will allow temporary nanopores in there so you can insert things in it. This is a picture of it. You can actually do 1 million cells per second, and those are some of our early designs. And these are all these microfluidic channels. In 2014, Scientific American named their top 10 breakthroughs in the world in every area. They picked CRISPR-Cas9 number 1 and squeeze what I just showed you number 2. I personally think they got it reversed, but thought it was okay. And part of why they did, again, coming from the paper, is you can look at the cell-penetrating peptide, nucleofection, which is electroporation, and then Armin's device, and you get much better. This is to try to generate iPS cells. You can also insert material into T cells and B cells and myeloid cells. And again, this is just untreated, no device in the device, and you can see that the device really just changes what you can get. In fact, Armin started a company on this. Again, that's Squeeze Biotech, and Roche already did a $500 million deal with them just to engineer B cells. This is, again, comparing it to electroporation with RNA. And again, with microfluidics, you do better than nucleofection. But one of the things that's exciting to us is that if you just look at viability, microfluidics does almost as well as untreated, and again, much better than electroporation, which is kind of the standard. And then on this slide, what I wanted to show you, which I think is really important and hasn't been looked at much, is most of the other methods, particularly electroporation, really changes the cell's nature. This is called the transcriptome, so this gives you messenger RNA. So if you look at a control, this is what it looks like. But if you look at what happens to the transcriptome in a microarray with electroporation, it's totally changed. Here's ours, which has changed a little, but not much. So I don't think the FDA, when they start looking at this, I think that will be a big deal in terms of how other ways of inserting things in cells can cause damage. And this is just the last slide. So far, Armin and the team have basically delivered things to 30 different cell types, everything they've tried, stem cells, primary leukocytes, immortalized cells, and they've tried 20 different types of materials, like proteins, nucleic acids, but even things like that are impermeable, like quantum dots and things like that. So anyhow, this is early, but it's already led to a company that's been funded by Polaris, and that Armin's actually running with the help of Amy Schulman, who's one of Terry Maguire's partners.